Alaikum wa rahmatullahi wa barakatuh. Dear students, I am Dr. Harra Sabah, Assistant Professor in Orthodontics in the University of Baghdad College of Dentistry. Our lecture today, entitled in Principles of Genetics, Genetic Factors and Inherited Factors, its role on craniofacial abnormalities. First of all, we have to talk about a basic information and definitions of genetics, and prior to start with more correlation between genetic and orthodontics. As we see here, in our words always we said that this baby resembles his father or his mother, but this word means many information for us and for the patient. So, the meaning of a human genome. An organism genome is defined as the complete set of genetic instructions for that organism. The human genome is made up of a double helix of deoxyribonucleic acid DNA, comprised of 3.2 billion chemical nucleotide base pairs, which is adenine, guanine, thymine, and cytosine. This genetic information is normally organized into smaller units ranging in length from 50 to 250 million base pairs for each, called chromosomes. As we see here, these are the chromosomes. The chromosomes is made up of a continuous stretch of the double helical DNA. These chromosomes made up of a double helical of DNA that is wrapped around proteins that are called histones. Altogether, we each inherit a total of 46 chromosomes, 22 homologous pairs of chromosomes called autosomes that are numbered by size and other characteristics, along with one pair of sex chromosomes that are homologous, which is XX in females, and only partly homologous XY in males. Looking closer at the chromosomes, they are further organized into smaller units called genes, which represents the smallest physical and functional unit of inheritance. A gene can be defined as the complete DNA sequence that codes for the synthesis of a specific polypeptide or the synthesis of a specific RNA molecules. As we see here, these genes situated here at each region of this chromosome is named locus A from the maternal origin, from the mother, and this is the other from the paternal origin. Within the human genome, we have to know what's the meaning of allele versus gene. Every gene resides in a specific location referred to as a locus. The term locus is used when describing a single genetic region or location, and loci is plural. Genes at the same locus on a pair of homologous chromosomes are called alleles. One allele would be a copy of the maternal allele and the other a copy of the paternal allele. As we see here, this is the gene and on locus A, as we see, and then, locus D, and this is gene allele 2, and if we see here, the corresponding one in the maternal origin is gene allele 2. So, when a pair of alleles are identical in DNA sequence, for example, allele A and allele A, from paternal and maternal origin, the individual is said to be a homozygous for that locus. However, when the two alleles have one or more differences in the DNA sequence, like allele A and allele small a, So the individual is said to be a heterozygous for that locus. Also, in the genetics glossary, there is genotype and phenotype. So what is the difference between genotype and phenotype? Genotype is a complete heritable genetic identity. It is a unique genome that would be revealed by personal genome sequencing. A genotype generally refers to the combination of alleles at a given locus within the genome, as in the previous exam, capital A and A from the maternal and paternal origin, or A capital from the maternal and A small from the paternal, or small a and small a from the maternal and paternal origin. According to the information gained in the Human Genomic Project, we now know that the human genome is about 99.9% identical from one person to another. Thus, there is only an estimated 0.1% variation within the entire DNA code between two people that makes each individual unique. So, in contrast to genotypes, phenotypes are the observable properties, measurable features, and physical characteristics of an individual. A phenotype is generated by summation of the effects arising from an individual's genotype and the environment in which the individual develops over a period of time. As we see here, this is the gene. Usually, the end product of the gene is the protein. So, this protein product is affected by environmental factors, and modifying genes. These modifying genes also produce another type of proteins that could affecting on the end results of the original protein, resulting in a specific phenotype, a characteristics phenotype for such person. Heritability of malocclusion. What is the methods of heritability of malocclusion? A trait is a particular aspect or characteristics of that phenotype. Examples of traits include eye color, hair color, mandibular jaw size, and stature. As we see here, there is a mandibular prognathism in the Habsburg family. This is royal family, have a dominant trait, which is a mandibular prognathism, as we see in this picture. And this family occupied for a long time this feature of mandible prognathism. Depending on the genetic influence on traits, the traits can be considered to be one of three types. Either monogenic traits, which is the Mendelian type, that trait develop because of the influence of a single gene locus. Polygenic traits, complex or common, the traits that are resulted from complex interaction of multiple genes. Or multifactorial traits, when polygenic traits are influenced by environmental factors along with the multiple genetic factors, meaning they are influenced by interaction of multiple genes, as well as environmental factors. So, in the heritability of malocclusion, the type of a trait that can be identified is named either autosomal dominant trait or autosomal recessive trait or X-linked recessive trait. The autosomal dominant trait, as we see here, that if the father is affected and the mother is unaffected, we can see that we can get an unaffected son with unaffected daughter and affected daughter with the affected son. In this type of autosomal dominant trait, there is half of the siblings would be affected and the other would be unaffected. While in the autosomal recessive trait, if the production of the trait or syndrome doesn't occur when only one copy of particular allele is present at the locus on a pair of autosomes, but doesn't occur when two copies of that particular allele are present at the locus of a pair set of autosomes. Two copies of the defective gene are required for the expression of the trait. The parents are heterozygous. The X-linked recessive trait, the recessive genes on X chromosomes express themselves phenotypically in males as if they were dominant genes because a male usually only has one X chromosome. Hemizygous. A male with the genotype is affected in the pedigree. So why we should a student of orthodontics be interested in genetics? The reason is very simple. Whatever affects the growth, development, and function of the oral and facial structure is of interest to the student of orthodontics. We have to know exactly why or how a malocclusion occurs, to what extent does it express in the next generation, what is its prevalence, and how will it react to a certain treatment plan, and most importantly, if it can be prevented or not. Mode of transmission of malocclusion. Malocclusion may be defined as a significant deviation from what is defined as normal or ideal occlusion. Many components are involved in normal occlusion, including the size of the jaws, the relationship between the two skeletal bases, the arch form, the size and morphology of the teeth, the number of teeth present, and the soft tissue morphology and behavior including lips, cheek, and tongue. So the dental and the skeletal characteristics that could be inherited by genetics. Most of the problems in orthodontics are not strictly the result of only genetic or only environmental factors, unless trauma, but a combination of both. It's important to understand the cause of a problem before attempting treatment. As we see here, and the studies showed that class 2 div 1 malocclusion appears to have a polygenic multifactorial inheritance. So environmental factors can also contribute to the etiology of class 2 div 1 malocclusion, such as digit sucking. Soft tissues can exert an influence on the position and inclination of upper and lower incisors, and the need to achieve lip-tongue contact for anterior oral seal during swallowing can encourage the lower lip retrocline the lower incisors, and the protruding tongue to procline the uppers, influencing severity of the overjet. So here, there is no purely genetic factor could be the cause for such classification and relationship of the upper and lower jaws. While in class 2 div 2 malocclusion, the studies showed that it have a strong genetic component and exhibits high genetic influence and is often considered as a genetic trait. Results of many studies suggest the possibility of autosomal dominant inheritance. Class 2 div 2 is a multifactorial polygenic complex trait, a number of genes acting additively rather than being the effect of a single controlling gene for the entire occlusal malformation. Also, high lip line, lip morphology, and behavior are also considered to be causing class 2 div 2 malocclusion. Furthermore, the presence of strong masticatory muscle pattern in class 2 div 2 cases is explained by the genetically determined muscular and neuromuscular system. As we see here in class 2 div 2 malocclusion, the upper incisors is retroclined and the upper lateral incisors is proclined. In the class 3 malocclusion, also many studies have suggested a strong genetic basis for mandibular prognathism, autosomal dominant trait. The genetic factors appear to be heterogeneous, with the monogenic influence in some families and multifactorial polygenic complex influence in others. As we see here, there is a superimposition of the facial profile photograph on the lateral cephalometric radiograph of a subject which is the son of his father. This is the son and this is the father. The son is aged 24 years old and the father 54 years old. The image showed that there is a mandibular macrognathism. The corresponding front and right lateral occlusal photographs, as we see here, reveal the more severe anterior crossbite of the son. Malocclusions associated with the genetic syndromes. Many craniofacial disorders and genetic etiology with the malocclusion including facial cleft, cleft lip and palate, Down syndrome, cleidocranial dysplasia, and osteogenesis imperfecta. All these syndromes associated with different types of malocclusion. Therefore, this is mainly due to autosomal dominant trait. Stable 1. The canine is the more stable tooth in the developmental field theory. So, according to this field theory, the local occlusal variables associated with the genetics, as we see here, firstly hypodontia. Hypodontia has a hereditary nature. Hypodontia means there is a congenital missing of single tooth. Hypodontia has hereditary nature. Maxillary lateral incisor is the most common tooth to be congenitally missing, next to third molars, followed by the second premolars. Hypodontia often exhibits familial occurrence and fits polygenic models of inheritance. Hypodontia and the reduction in tooth size are in fact controlled by the same or related gene loci. So as we see here, the most commonly missing tooth is the lateral incisor. Also, supernumerary teeth, most frequently seen on premaxillary region, also appear to be genetically determined. Mostly mesodens are more commonly present in parents and siblings of the patients who exhibit them. The mode of transmission could be explained by a single autosomal dominant gene. Tooth size and shape. Studies have shown that tooth crown dimensions are strongly determined by heredity, as dietary habits in humans adapt from a hunter-gatherer to a defined food culture. Evolutionary selection pressures are tending to reduce tooth volume, which is manifested in third molar, second premolar, and lateral incisor, according to the Buller's field theory. Ectopic maxillary canine. Various studies have indicated a genetic tendency for ectopic maxillary canine, being one of the anomalies in a complex and genetically related dental disturbances, often occurring in combination with the missing teeth, microdontia, supernumerary teeth, and other ectopically positioned teeth. So the clinical implications of genetics in orthodontics, that malocclusion with a genetic cause is generally thought to be less responsive to treatment than those with an environmental cause. The greater the genetic component, the worse the prognosis for a successful outcome by means of orthodontic intervention. However, knowing exactly the relative contribution of genetic and environmental factors is not always possible, malocclusions of genetic origin, skeletal discrepancies, when detected in a growing period, are being successfully treated using orthopedic and functional appliances, except in the extreme cases where surgical intervention is required. And thank you for your listening.